Today's video is foundational concept number 1 of biology, 1A, proteins and amino acids, structure and function. So we're continuing with the second video on classifications of amino acids and whether they are acidic or basic. So first we're going to talk about protonation and pKa. So when the pKa is higher than the pH, the group is protonated. When the pKa is less than the pH, the group is deprotonated. When pKa is the same as the pH, half of the group is protonated and half is deprotonated or neutral. So protonation is very important when you're looking at acidic and basic amino acids. And I talk about it as protonation rather than a charge because when groups are charged, they may be either protonated or deprotonated. And they may have a positive or a negative charge. So first we'll look at the amino acid backbone's protonation. So in the human body at a pH of around 7.4, the amino acid backbone has an overall neutral charge because the carboxylic acid group is deprotonated, giving it a negative charge, and the amino group is protonated, giving it a positive charge. So in the picture down on the right, you can see the amino acid backbone, and this is called a Zwitter ion form, which we will talk about in another video. The pKa of the carboxylic acid group in the backbone is around 2, which is really important to remember for the MCAT. Therefore, to be protonated, the solution must be lower than a pH of 2. So as you can see on the left, this is representation of this carboxylic acid being deprotonated at a pH of higher than 2. So it has a negative charge. And on the right, you can see it protonated at a pH of less than 2. And it has a neutral charge. The pKa of the amino group in the backbone is around 10. Therefore, to be deprotonated, the solution must be higher than a pH of 10. So on the left, it's a representation of the amino group in its deprotonated form with a neutral charge. And on the right, it is it in its protonated form with a charge of positive 1. With basic and acidic amino acids, because of this deprotonation and protonation, the acidic and basic amino acids are the only amino acids that are charged at a neutral pH within the body. So these pKa values must be memorized as their values are not often given on the MCAT. And in general, these are going to be the only amino acids that you're going to be tested on for pKa values. So both acidic and basic amino acids are known as polar amino acids. They are hydrophilic and they want to interact with water through hydrogen bonding. They usually are on the outside of a protein or bound to the outside of a cell membrane for interactions. So we will talk more about what polarity means and hydrophilic means in another video. Acidic amino acids include aspartic acid and glutamic acid. So that is what they're called in their neutral form, but within the body they are known as aspartate and glutamate with their -1 charge. Their side chain pKa is around 4, and their R group has a negative charge when the pH is higher, so like I said, in the human body, and at a neutral charge when their pH is lower. Aspartic and glutamic acid both have a carboxylic acid group, which makes them acidic. So in this image, you're looking at them in their neutral charges because they're both seen at a pH of 3, for example, where the amino acid backbone is still in a Zwitter ion form, so a neutral, but there are hydrogen atoms added onto the side chain. So most often on the MCAT, you're going to see amino acids in their ionized form because this is them at the body pH of 7.4. So here they are as glutamate and aspartate with their -1 charges. As you can see, they're both missing their hydrogen ions in the side chain. So some easy ways to remember the difference between these two amino acids is glutamate is a glutton, so it has an extra CH2 group. Aspartate is more acidic, there's less stabilization, and that is because there's only one carbon group before it, whereas glutamate has 2. Both of them have a carboxylic acid that makes them polar and able to be phosphorylated. So we're going to talk more about phosphorylation when we're talking about amino acids that are more commonly phosphorylated. Aspartate sounds like date, so you think of the shorthand being D. It's the easiest way to remember it. The glutamate shorthand is E, which comes after D, similar to A coming before G in the alphabet. So when you think about trying to memorize these, I have little ways. Acid puts you in the ED, so acid puts you in the emergency department, right? And ED stands for glutamate and aspartate. Okay. So now we're on to basic amino acids. So the basic amino acids are lysine with a pKa of 10.5, arginine with a pKa of 12.5, and histidine with a pKa of 6. Their R group has a positive charge when the pH is lower than their pKa. It has a negative charge when the pH is much higher than their pKa. Again, I'm just going to stress the importance of trying to memorize these pKa values. So it is important to remember that arginine is the most basic amino acid, and that histidine is only partially basic because it has a pKa very close to neutral. So first we'll talk about lysine. So lysine shorthand is LYS or K, which is kind of hard to remember. It has an amino group, which makes it basic. So in the body, this group becomes NH3+, just like in the amino acid side chain. So an easy way to remember it is thinking of the structure as a line. So on the left, you see a pH of 1, lysine will have a positive 2 charge because both of the amino groups are protonated, and as is the carboxylic acid group, making it neutral. The pH of 7.4, it has a positive 1 charge because the backbone is neutral, and the amino group is protonated with a positive 1 charge. At a pH of 12, lysine is in its Zwitter ion form, isoelectric point, meaning that it has a neutral charge. Arginine has 2 amino groups, which make it basic. So the way that I remember the structure is that arginine has 2 ends in the name and 2 ends in the chain. So on the left-hand side, you can see at a pH of 1, it has an overall positive 2 charge. At a pH of 7.4, it has a positive 1 charge. And on the right, at a pH of 14, it's going to have no charge because its pKa value is 12.5. So histidine, its nitrogen, is located in a stable ring, which gives it a partial positive charge at the body's pH. So the way that I remember the structure is thinking of histidine of having 2 ends in the R group. So I highlighted the Di because di means double. You'll often find that Di when we start talking about chemistry and ring structures. So on the left, you see histidine in a pH of 3 because it's going to have a positive 1 charge. And then on a pH of 7.4, it's going to have a 0.25 charge. So it is just mildly basic. So just in a quick summary of the basic amino acids, all of them have nitrogen in their R group. Basic amino acids have a positive charge at the body pH. And some easy ways to remember the shorthand for the amino acids is that lysine is the last L amino acid alphabetically. So it's represented with a K. Arginine sounds like a pirate saying R, and histidine shorthand begins with H, so it is quite easy to remember.